Welcome to Dr. Balaga's God Knowledge Doc podcast, your quick dive into the latest medical breakthroughs to keep you ahead of the curve. Let's get started. Today is October 19, 2025. This video is on an article published in Cell Death Discovery on 6 October 2025, a Nature journal. The video is on a review article titled The Role of Cellular Senescence in Cardiovascular Disease. What is cellular senescence? It's a stable form of cell cycle arrest with altered morphology, metabolic activity, and secretion of inflammatory factors. How does it relate to aging? Senescent cells accumulate with age, contributing to tissue dysfunction and chronic diseases like cardiovascular disease. What triggers cellular senescence? Telomere shortening, DNA damage, oxidative stress, mitochondrial dysfunction, oncogene activation, and inflammatory cytokines. What is SASP? SASP is senescence-associated secretory phenotype, and these include pro-inflammatory cytokines, chemokines, growth factors, and proteases secreted by senescent cells. Is senescence reversible? While cell cycle arrest is permanent, some features like SASP or senescence-associated secretory phenotype can be modulated therapeutically. Senescence in cardiovascular pathologies. How does senescence affect high blood pressure? By promoting endothelial dysfunction, oxidative stress, arterial stiffness, and increased peripheral resistance. What is its role in atherosclerosis? Senescent endothelial and smooth muscle cells promote plaque formation, inflammation, and instability. How does senescence contribute to myocardial infarction? Senescent cardiomyocytes and progenitor cells repair, induce fibrosis, and limit regeneration post-myocardial infarction. What about heart failure? It accelerates cardiac fibrosis, myocardial dysfunction, and autophagy impairment, worsening heart failure progression. How does senescence affect cardiac arrhythmias? Or does this senescence affect cardiac arrhythmias? Yes, via ion channel remodeling, electrical conduction impairment, and atrial fibrosis leading to atrial fibrillation. Molecular mechanisms and dual effects. Which pathways regulate senescence? Key players include p53, p21, p16RB, mTOR, sirtuins, SIRT1-6, NF-kappa B, and reactive oxygen species DNA damage signaling axes. What are senolytics and xenomorphics? Senolytics eliminate senescent cells, example dasatinib, ABT263. Xenomorphics suppress SASP without killing the cells, example rapamycin and metformin. Can senescence be protective in cardiovascular disease? In early injury phases, SASP mediates repair and limits fibrosis. In chronic phases, it promotes maladaptive remodeling. What is the impact of senescent heterogeneity? Senescent cells differ by tissue, subtype, and function, affecting how therapy should be tailored. How do metabolic and epigenetic factors contribute? Decrease NAD+, RNA dysregulation, and DNA methylation foster a self-reinforcing loop of aging and cardiovascular disease. Which drugs target senescence in high blood pressure? Rapamycin, SIRT1 activators, example resveratrol, NAD+ precursors, example nicotinamide ribose, and ACE inhibitors show benefit. What are emerging therapies in atherosclerosis? Senolytics like ABT263, SIRT1 or SIRT6 agonists, glutamines inhibitors, microRNA 217 and SIRT6 modulation reduce plaque burden. What helps post-MI remodeling? Venetoclax, SIRT1 enhancing MSCs, ABT263 PLGA, and nanoparticles to improve angiogenesis and reduce fibrosis. How does targeting senescence help in heart failure? SIRT1-6, PGC1 alpha activators, and p53, p16 inhibitors improve mitochondrial function, diastolic performance, and reverse fibrosis. Any role in cardiac arrhythmia management? quercetin and dasatinib mitigate atrial fibrosis and reduce vulnerability to age-related arrhythmias. Research frontiers and open questions. Do all senescent cells behave the same? No. Subsets, example, pro-inflammatory versus reparative, respond differently, complicating uniform treatment strategies. Can senescence clear harm tissue repair? Yes. Removing too many senescent cells may impair regenerative cues or immune modulation. How can we personalize anti-senescence therapy? Via integration of single-cell multiomics, epigenetics, and spatial transcriptomics to identify cell-specific vulnerabilities. What are the clinical challenges? Balancing efficacy and safety, off-target effects, and demonstrating long-term benefits in trial. Is prevention feasible? Potentially, through lifestyle interventions, especially NAD+ preservation and antioxidant strategies to delay senescence onset. Some eponyms in cellular senescence research. Leonard Hayflick. He discovered the Hayflick limit, showing normal human cells have a finite capacity to divide. Gottfried Schatz contributed to understanding of mitochondrial dysfunction's role in aging and senescence. Judith Kempisi pioneered in biology of senescence and its link to cancer and age-related diseases. Jan Wick advanced the concept of genomic instability as a hallmark of aging. David Sinclair, known for work in sirtuins and NAD+ aging and cardiovascular health. If you enjoyed this episode, share it with your colleagues. When we change how we see disease, we change how we shape lives. Until next time, stay curious, stay compassionate, and keep seeking wisdom. Podcast brought to you by masterbedfacts.com. This podcast is for physicians and healthcare providers. If you enjoyed this video, please like, subscribe, and forward to your friends.